Patricia Demore, a professor in Stephen's Eye Institute in Harvard Medical School. Professor Demore is going to be coming to us over Zoom. So hopefully, yes, you're there, Patricia, I can see you now. Can you? Yeah, great. We'll let you fire ahead when you're ready.
Okay, thanks. Can you hear me now?
Yeah, we can hear you.
Great. Well, thank you everybody for your patience and having me join you by Zoom. I had intended to be there, but had a family issue come up, so hopefully this will be close to the effect that I had when I was there. I'm also, going to leave time at the end and say right now that anybody's welcome to reach out to me at any time. So I have a little bit of an off-the-wall topic here. This is not a topic that you generally hear about probably in any session whatsoever. And it's an area that we got into probably about 5 years ago. So let's see, let's make sure this is working. So disclosures, none of these really have anything to do with this talk. There's very few people are interested in the glycocalyx right now, unfortunately, including study sections, but it's a different story. So, let's see. So, glycocalyx, if you trained in biology at any level, I'd say undergraduate, this is a term that you heard about at one point. And so, it actually means calyx is a coat and glyco is sugar. So, it's a sugar coating, literally. And this is a great picture from the literature, not mine. We've been trying to reproduce something like this. I'm not saying it's not reproducible, it's just very tricky. You have to perfuse fix and then stain specifically with a particular dye. But what you can see here on the surface of the endothelium, so when we say glycocalyx, we're talking about the vascular endothelium. You can see this, it almost look like hairs, and again, that's due to the nature of the fixation and staining. But it is a very complex meshwork. I actually like to think of it in some respects as the extracellular matrix of the apical part of the endothelium. We all know about the basal extracellular matrix, and I think of this in some respects as the apical extracellular matrix. I don't know how you're doing this, John. Do you take questions as you go along? Because I'm happy to answer any questions. I don't want to leave anybody behind in anything that I say. So you can,
We'll take questions after.
Okay, after. Okay, so what you see here is the glycocalyx, and the way I'm going to structure this talk is probably the first 2/3 of it is going to be general about the glycocalyx, things that maybe you remember and maybe you don't remember, but intended to give you a sense of what the known functions of the glycocalyx are, because there are actually quite a few known functions. And then in the last 10 minutes, I'm going to talk to you about sort of unanswered questions and the sort of the big remaining issue, not remaining issue, but the big issue is what's the relative contribution of each of the components? As you'll hear shortly, this is a really complicated and complex structure. And the sizes are here because it will become evident later on that these adhesion molecules, things that you're very aware of, things like PCAM and ECAM and selectins, are basically embedded within this very complicated matrix that I'll talk to you about the components shortly. So you can already see that because of the length of this, and what I'll tell you soon is it's very hydrated, is that this one of the major functions of the glycocalyx is it provides protection and hides these adhesion molecules that otherwise would initiate inflammation, which is certainly what we don't want. We don't want unintended and sort of inflammation that doesn't have a purpose. So that's what we're going to be talking about. So I'm going to talk about, at this point, some of the functions that we know about. Again, these have come up over the years and are now taking on some additional relevance. So it has obviously a barrier function. I showed you that. You could tell just by looking at the prior slide that a mesh like that is going to act as a barrier. And then depending on the components, we'll talk about those again in a minute, but they have, depending on where they are in the vasculature, they have very different sizes and very different charges, and that's all determined by the sugar side chains. And based on those, they have variable barrier functions. In fact, there was just a very big paper maybe about 3 or 4 months ago, I think it was in Science or one of the big high-impact journals, about the role of the glycocalyx in the blood-brain barrier. So it's actually starting to be more acknowledged, the contributions of the glycocalyx. I mean, when I think of this, I think back to the early days of studying the vasculature. In the very early days of vascular biology, before most of you were born, it was that people thought that the capillary or the vasculature was a tube, and it was lined by endothelium, but they were inert. And so early papers called it a layer of cellophane. And so I would say the glycocalyx is about a little bit beyond that at this point. It's known to be there, but the wide range of potential functions is just beginning to be uncovered. A very well-known function is mechanotransduction, and it actually senses shear stress from the blood. And this is one of the functions for which the molecule is actually known. So heparan sulfate side chains can actually sense sense shear stress. Some of the components of the glycocalyx are actually attached to the cytoskeleton, and in that way can actually transduce mechanical forces, and those can get translated within the cell into biochemical signals. So I'll point that out later on when I talk about the specific molecule that we've been talking about. I wouldn't say mechanistically that's understood incredibly well, but it certainly is known to be a mechanotransducer. And then the very big function that I've already suggested is as an anti-adhesive. So that meshwork, that tall hydrated meshwork, actually is, as I spoke about, a physical shield that prevents leukocytes and platelets from adhering to the endothelium. The endothelial surface, it can be very pro-inflammatory under certain circumstances, and actually there's a good number of adhesion molecules that are expressed on the surface of the endothelium, even under quiescent conditions. And so when I say quiescent, I mean non-inflammatory conditions. Why would that be? Well, you want the vasculature to be ready to respond to inflammation when necessary. And this meshwork is there, and as I'll talk to you shortly, can be shed really quickly. It's shed by things like atoms and downstream of inflammatory molecules. So the entire vascular glycocalyx can be shed, and in fact, that's a hallmark of sepsis. So when you think about sepsis or long COVID or cytokine storms, that's what's happening. You have significantly high circulating levels of things like IL-1 beta and interferon, and that can shed the entire glycocalyx surface. So if you actually look into the literature, a major part of the literature on glycocalyx has to do with the fact that it's so completely shed during a cytokine storm. And you can imagine what happens then. I mean, you've all thought about it in terms of long COVID. If you shed the entire glycocalyx, you've revealed all of the quote-unquote previously hidden adhesion molecules, and that can lead to systemic, right? That's what sepsis is, systemic inflammation, which is just a vicious cycle. So in that regard, the glycocalyx plays an extremely important protective role. It also regulates coagulation. The side chains contain heparan sulfate, and then, as we'll talk about in a moment, that mesh includes transmembrane proteins, surface-attached proteins, and also plasma proteins that just get sort of entangled and trapped or otherwise bind to the matrix. So it's a place where, not unlike, if you think about it, the extracellular matrix, the heparan sulfate molecules that are part of the glycocalyx can trap a lot of plasma proteins. And you probably all know, and we'll talk about in a bit, heparan sulfate is a molecule that, because of its charge, binds lots of things, growth factors and many circulating molecules. So it's also a reservoir in some respects. And therefore, because it holds it away from the surface, it limits coagulation. And this is what I was just talking about. It's a reservoir for plasma proteins, just as, frankly, in some respects, the extracellular matrix is. So it binds and sequesters protein that also binds a lot of growth factors. You know, the FGFs, VEGF, all of those, PDGF, all of those growth factors can bind heparan sulfate. In fact, in many cases, that's how they were originally purified. And you can see it also binds SOD superoxide dismutase, which means you have a concentrated antioxidant at the cell surface. It also stabilizes oncotic pressure by trapping those proteins, and as I said, binds other important plasma proteins. During inflammation, it regulates vascular permeability, and this is what I spoke about a bit before, that once you shed the glycocalyx, and that can be done very locally, right? You can have a very local inflammatory response so that the glycocalyx is just going to be shed, say, in the area of a wound. So that once it's shed, and that's, again, a topic of a lot of interest, is understanding the shedding, because if you can control the shedding, you can actually rein in sepsis. Once that is shed, it's sort of a vicious cycle. You've let the horse out of the barn, as it were. And this is what I spoke to you about before. These are constitutively expressed molecules, and once you lose the glycocalyx, you've, exposed what's sitting there. And then remember, the cytokines that are actually leading to, say, atom or enzyme production that sheds the glycocalyx is at the same time upregulating additional adhesion molecule production by the endothelial cell. So you can see how this is going to lead to a very dramatic and potentially damaging systemic inflammatory response. It also impacts, this is one of the earlier known functions, because it maintains what they've called an exclusion zone. It keeps red cells in the center and prevents them from sort of sludging along the edge and keeps flow going. So just what this says, helps reduce resistance and prevent sludging, particularly in capillaries, because you all know that, red cell is, basically the size of a capillary lumen. So if you don't sort of grease the surface of the endothelium, you could have unnecessary and probably damaging resistance. So those are the known functions. Other things to know here are that the glycocalyx is really variable. It's not a single structure, just like the extracellular matrix. It's not a single structure across the entire vasculature. It varies throughout the vascular tree. And again, these things that I'm pointing out are the tip of the iceberg. You can certainly go and read more about these, but I want to give you a sense of the variability of the glycocalyx. So the artery has probably some of the thinnest compared to microvessels. You can already jump down and see that the glycocalyx at the level of capillaries and probably venules is the most critical location. Arteries, it's a thinner, and that's where shear stress happens. And in fact, the molecule I'll talk about later, we've shown is downregulated by shear stress. So it may be that the shear stress is actually what's leading to downregulation of the molecules. Veins have low shear stress, so they have less developed matrix. That's not where you're going to have inflammation happening. Arterioles, it's relatively thick. Again, those are resistance vessels, so you're going to have, you're going to want to have response to shear stress and that's where mechanotransduction happens. In capillaries, it's very thick, and we've talked about why. It's important for vessel diameter, keeps the blood flow. It also helps to maintain oncotic pressure. We're not going to talk about it. That's a different lecture. And then in the venules, it's supervised for immune surveillance, as we've talked about, because it prevents leukocyte adhesion, but it can be shed, as I've pointed out. There's also specializations in different tissues, so I'm going to just go over these quickly so that you know, again, as you'd expect, the vasculature is specialized for each tissue, so the glycocalyx is going to be specialized. So in the lung, it's thick. It helps to maintain the alveolar capillary barrier. In the kidney, it's very dense. We've looked at that ourselves because why? There's a lot of filtration that happens there. In the brain, it's actually integrated into tight junctions. Again, I refer you to the paper that was published a couple of months ago, invoking the glycocalyx in the blood-brain barrier. And then the sinusoids are an exception. Why? Because that's a place of very high flow, and flow in and out. Liver is a major place of production of plasma proteins, and also a major site of metabolism. So you don't want that access to be slowed down in any way by the glycocalyx. And then what are the components? This gives you, again, just a sense of what the components are. So there are membrane proteoglycans, membrane-bound proteoglycans. You've heard of many of these, the syndicans, the syndicans and the glypicans. And I'm going to talk to you about endomucin, which is the molecule that we've been studying. I'm going to give you a few minutes on that just to give you a sense of what's to be learned about these individual components. And we know a good amount about syndicants. It's one of the major sites of heparan sulfate side chain, so where a lot of the heparan sulfate action happens. And then the side chains are where a ton of variability comes into it. And I should point out that it's really difficult to study the composition of these, and you can see why. We have great mechanisms for studying sequences of RNA and DNA as well as proteins, but the technology for basically sequencing sugars is way behind. And so there's a lot of sort of technology that needs to be developed there before we can go into a tissue and say, oh yeah, here's the profile of the proteoglycans or the GAG side chains, because it's these side chains that are going to determine the charge, also what they bind. And I point out, I've mentioned heparan sulfate a lot, but for good reason. It's a major component, and again, it binds lots of things. So it's really a great site for sequestration. These are lesser contributions, but again, we don't know everything that we need to know about what these various GAG side chains are doing. And I'll talk in a minute about what we're trying to figure out. Very difficult. You basically have to use mass spec at this point to try to figure it out. Pablo Argueso's lab is taking a different tact. He studies ocular surface, say mucins, and he's going in trying to understand synthesis. So that's a complicated one. These are multi-step synthetic pathways. Other things that are in the glycocalyx, heavily glycoproteins that you're well aware of. I'm not going to go into all of these because all of these are familiar to you. You've heard lots about integrins. That's part of the glycocalyx. Mucins are those very tall. In the first picture that I showed you, the tallest structures are probably mucins. They're heavily glycosylated, O and N glycosylated, and they tend to stand up straight. So they're a major source of sort of the barrier or steric hindrance provided by the glycocalyx. These are giant. These can be very big. These can be extremely big and are, just like found in mucus layers, but, have been well studied on the surface, again, by Pablo Argueso and Eileen, studied that, and have many varieties and a lot to be learned in that area. And then other things that I described, plasma proteins that can get taken out of as they circulate by and then get enmeshed into the matrix. So what could those be? long lists. Albumin, which is, as you know, involved in, regulating the oncotic gradient. This is where the anticoagulants come in. Lipoproteins can get stuck there. Again, I've referred to this a lot. I've referred to it a lot because a lot of my career has been spent studying growth factors that bind to heparan sulfate. So this one is close to my heart. But also, think of it, it does provide a local source of growth factors that can be accessed when needed immediately. And then varieties of enzymes are also held there. Hyaluronic acid is a non-sulfated gag, but is very important in hydration. So those mucins tend to have a lot of hyaluronic acid associated with them because that helps them stay hydrated and provides the meshwork some of its solid structure as you would. So, John, how much time do I have left? Okay, never mind. I'll figure it out. So this is what I want to get into in the last few minutes, is to talk about known structures. So as I've said, when people talk about the glycocalyx, they tend to talk about it as a structure. And so they'll say the glycocalyx is a barrier, or the glycocalyx, regulates this. But not much has been done about the individual components. So if you want to try to understand, what's happening in the glycocalyx, for instance, it probably plays a major role in pathology, but very hard to sort of parse that because we don't, as I said, have a good way to try to figure out how does it change. We can certainly look at thinning. So you can look at thinning by, either staining, which is sort of very semi-quantitative, or you could do what we're trying to do now, which is you prepare the membranes from mass spec and you try to quantify the various side chains. But if you really want to try to figure out what's changing in pathology, you need a way to try to sort of quantify the kind of changes that are happening instead of just saying the glycocalyx thins. Are certain parts of the glycocalyx thinning? And in pathology, do certain parts get overproduced? All of that are big unknowns at this point. So in the last few minutes, I'm just going to talk about this one molecule. It's work from my lab, but just as an example of what is potentially there to be studied in the glycocalyx. So we came across this molecule. I mean, we didn't identify this molecule, but it was known. People use it as a marker. It's a spectacular marker for capillary endothelium, if you need a good marker. But we came across it in a particular way, which I can describe. I don't want to take the time now. It's actually considered a mucin because of all this O-rich glycosylation, but it's quite small compared to mucins. As I said before, many mucins are in the millions. This is just 150 kD when it's glycosylated. It's got a transmembrane domain and potential phosphorylation sites. I say potential because we've been studying this for a while and we have not yet identified a function for these. So I'm just going to give you maybe a few slides of data to show sort of the kind of project or program that can be developed around a single glycocalyx molecule. In fact, we don't even know what percentage, say, of the glycocalyx in capillary endothelium this represents. That's even hard to find out. So it would be nice to know that, but we don't know that yet. Antibodies aren't going to help. They have variable affinity. So, is this 2% or 20%? No idea yet. Again, it's a good marker, and here's evidence of it being a good marker. Interestingly, you can see it's expressed by veins, right? This collapsed structure with no real media. Another one here, and all of the capillaries. But here you see an artery, which you can tell is an artery because why? The surface is thrown into these waves by the contraction of the surrounding media, and it's not expressed there. And as I alluded to earlier, in a separate series of studies, we've actually shown that endothelial cells subjected to arterial shear stress downregulate VEGF. They actually do downregulate VEGF too, but they downregulate endomucin. So endomucin is pretty much gone here. If I took these cells and put them in culture, they'd express endomucin because the shear stress would be gone. So if you study arterial cells in culture, they'll have endomucin because they don't have any shear stress. So, and what's the signal that's picking up that? Maybe heparan sulfate. We haven't looked yet. So that gives you an idea of what the very unique distribution is here. We studied it in the developing mouse vasculature. I'll just point this out, which is very cool. Here, this is a primordial vascular network. It's everywhere, right? But by day 7, when you're remodeling the retinal vasculature, flow has started, shear stress has started, and the expression that used to be in all of the vessels is now gone in the artery. A really clear demonstration of the effect of shear stress, downregulating a component of the glycocalyx. Importantly, very highly expressed at vascular tips. That plays into the fact that, you know, we've identified a new role. One thing I can show you is if we inject SI endomucin into the growing front of a retinal vasculature, you know, you can inject into the pup eye, and you can see it totally disrupts the vasculature. So based on that, we thought and we suspected it was involved in signaling. And I'm going to show you 3 or 4 slides just to show you a new function of a glycocalyx molecule that hasn't been identified. We first showed that if you knock down endomucin, this is a classic assay that you probably all know, grow endothelial cells to confluence, draw a wound, and let them heal. If you add VEGF, you get all this healing. This is where it started, this is where it is. But if you knock down endomucin, it can't heal anymore. And interestingly, if you overexpress endomucin, so you see how much you've blocked healing, if you overexpress it, you improve healing. So endomucin is influencing VEGF signaling. And this is published, so I'm going to just go through it relatively quickly, but so just to show you, this is a component of the glycocalyx. We saw similar results with proliferation and tube formation. That's all been published. And what we found, I'm going to tell you the take home, and then I'll show you a few slides of data. Endomucin on the cell surface of the endothelium is necessary for VEGF-R2. So VEGF-R2 binds, associates with endomucin, and for some reason, which I'll tell you, the endomucin facilitates VEGF-R2 getting internalized into the cell after it binds VEGF and signaling. And if you don't have endomucin, the VEGF can get phosphorylated, VEGF-R2 can get phosphorylated, but it won't signal. VEGF-R2 has to be in the cell and transverse the cell whereby it continually signals. And if you just stick it on the cell surface, you get it stuck on the cell surface, it doesn't signal. That's where the inhibition comes from. So this is just to show you, this is just a way to trace the VEGF-R2. And you see if you add VEGF, and you follow, if you don't add any VEGF, you just add BSA, we've washed the unbound VEGF-R2 off this VEGF off the surface. You see there's not much internalized. If you add VEGF, you've internalized all this receptor, just as you'd expect, because under these circumstances, the VEGF's doing its job. And this shows you get a huge increase in VEGF-R2 internalization, just under normal circumstances. But if you take out endomucin, and we can do that very effectively just by knockdown, you can see that you've prevented VEGF-R2 from being internalized. There's no difference. So here's all the internalization. This is just under normal conditions. But if you knock down endomucin, you can't internalize R2, and that's quantified. And this is just a way to show that biochemically, so that people like to see the biochemical. You can see that when you add VEGF, this goes way down because the R2 is disappearing from the cell surface into the cell, right? But we're isolating membranes here and basically asking, is the R2 disappearing from the cell surface, as it should? So when you add VEGF, the R2, under normal circumstances, right, regular endomucin, the VEGF-R2 disappears from the cell surface. But if you take endomucin away, you can't internalize VEGF-R2 anymore. And that's why you don't get the biology. And what's the reason? This, again, that's quantified. We've shown that VEGF actually interacts with VEGF-R2. I'm not going to go through all that. You can, again, it's published. You can take a look at it. So you can see that it actually interacts. The key is, and I'm not going to go through this slide in great detail, what happens is the endomucin actually binds to AP2, which is the complex that's required for VEGF-R2 internalization. So all this to say, VEGF-R2 on itself cannot recruit clathrin. If it can't recruit clathrin, it can't be internalized in a clathrin-coated pit, and it can't continue to signal to do its job, get you biology. Therefore, in this case, what's happening is endomucin is coming along, and it's the endomucin that's binding the clathrin, basically for the VEGF-R2, and allowing the clathrin-coated pits to form and for the VEGF-R2 to be internalized. And so basically this is what shows that endomucin is interacting with the AP1 and AP2. So it's very cool, I think. I mean, the fact that R2 in and of itself cannot recruit clathrin for the important internalization step. Lastly, science-wise, this is just 2 slides. There's a whole story about this. When I said that you need glycocalyx to keep the endothelial cell surface from undergoing sort of spontaneous inflammation or unwanted inflammation, we did this. I'm just showing this very simple slide. So as you probably know, one way to look at leukocyte binding to the endothelium is this system. I'll just show you the system. We did this with Pilar Alcaide. In this case, you grow Huvix, but you could grow anything on this here. And then you put a gasket and flow through it, and then you flow neutrophils by it. And if the surface of the endothelium is inert, as it should be, not really inert, but non-activated, the neutrophils are not going to stick. But if it's somehow activated, or not just activated by cytokines, but if for some reason some of the adhesion molecules are exposed, you will get neutrophil sticking, and that's what we saw. So this is, if you put scrambled siRNA there, these are the endothelium, and there's a couple of neutrophils sticking there. But when you take endomucin away, you get tons of neutrophils sticking. And we went on to show that this is basically binding of the neutrophils to ICAM. I didn't bring those data because, again, they're published. But that means this single molecule, endomucin, which I've sadly pointed out, we are not, don't even know what percentage of the glycocalyx it provides, is sufficient to prevent this kind of leukocyte binding to the endothelium. That's just one molecule out of the many that I've described. So I'm going to tie this up by saying that while the glycocalyx plays a number of known important roles, it surely has additional functions that await identification. Unanswered questions, there's many, many. What are the, as I've alluded to, what are the relative contributions of the individual glycocalyx components? What are the additional functions of the glycocalyx? How is it altered in ocular pathologies, including dry AMD? Well, why would I say dry AMD? Because there is, if the glycocalyx is degraded for some reason, for instance, we already know we did a knockout animal, and when we knock out endomucin, the choriocapillaris goes away. Why? Because the choriocapillaris is dependent on VEGF-R2 signaling. There's a couple of papers out there talking about changes in the glycocalyx in diabetic retinopathy, and somebody actually went so far as to try to replace it. So these are very sort of, I would say, early experiments in trying to figure out what's going on pathologically. Very little knowledge about that so far. And is it targetable as a therapeutic? I say that because there's that story that I just mentioned where somebody tried to replace the glycocalyx in a diabetic retinopathy model and, reported some positive effects. So this is just a small selection of future readings. I can provide more, but, you can PubMed glycocalyx and find lots out there. So I'm going to stop there, and I'm happy to answer any questions.
Thank you, Pat. This is a very nice talk. Thank you. So, questions? I understand you. So you have a knockout endomucin?
Yeah, we've done a knockout that was a global developmental knockout, and we're working on an inducible one right now.
So it's viable, the global one, because the no VEGF?
So,
that's surprising,
yes, exactly. No? Yes, I was shocked and disappointed. And just no. So, right? So if we think that VEGF signaling is so important, and we know from the literature that you knock out VEGF and you have an embryonic lethal at day 10 because you don't get an aorta, our thought was that there could be redundancy. That's what everybody says who has a protein that they love that turns out to not show a big phenotype. But it is true that there's a lot of genetic redundancy, particularly in signaling pathways. So if you're doing it globally, my theory is you've given it the opportunity, or the organism, quote unquote, the opportunity to invoke a redundant pathway, and that may be the case. The phenotypes that we saw a significant inflammatory response in kidney. We see choroidal thinning. We see delayed vascularization in the retina, but not a knockout. So I think what we're working on right now is trying to knock it out around term in pups when vascularization just starts. So the theory there would be if what's happened is there has been redundancy, if we allow the redundancy to sort of take place in early development, but then knock it out just around term in the mice, we might see a more dramatic impact on vascularization of the retina and also on other, tissue vascularizations, because a lot of tissues have a good deal of vascularization postnatally. But it's a good question, and we were surprised.
Thank you. Yes.
Ernst Damm from the University of Regensburg. Is there a difference in the glycocalyx between fenestrated capillaries, like in the choroid, or in the retina, where you have a very tight barrier?
I would say there is. Nobody's actually looked at that. But you would imagine that. I'm going to speculate because there's no data that in a fenestration, like a sinusoid to some extent. Remember that I said that sinusoids have almost no glycocalyx. So if you consider a fenestration, a sinusoid light, if you will, then you would expect that there would be less glycocalyx there, whereas blood-brain barrier has a lot of glycocalyx and solid. One thing I can say certainly is blood-brain barrier has more than a fenestration for sure. But in terms of, quantity measured and quality, what constitutes it, not known.
Pat, this is John. I have a quick question. We had a beautiful talk by Rob Mullins talking about the accumulation of complement activation in the RPE and the capillary ghosts that form. Is there any evidence that this early activation of this cascade is causing the shedding and activating the atoms and causing the shedding and contributing to that?
No, there's not, but I would not be surprised. When we talk about inflammation like sepsis, we're talking about frank, inflammation such as one sees in wound healing. When we talk about inflammaging or parainflammation, the words that we come up with, we have very little understanding of what's happening locally under those circumstances. So I can tell you that if we add very high levels of, say, IL-1 beta or interferon to endothelium, we can pull the whole glycocalyx off. But if we go way back and much lower, a hundredth of a dose, we can see lesser of an effect. So my sense would be if you are getting local inflammation, which I believe you are, that you are eating away to some extent at the glycocalyx. But it's another unknown question to add to the list. But that's always been my guess, is that you are impacting it because you're then making that vasculature more prone to, neutrophil adhesion. I mean, think about in diabetic retinopathy. You know, it's been shown that neutrophils are sticking and clogging capillaries in early diabetic retinopathy. Well, why would that happen? It might be because you've eroded the glycocalyx there, making it easier for a leukocyte or a neutrophil to be sticking and clogging the vasculature. That's easier to see than, right? So Jerry Luddy did those studies, some of those studies. You could see in humans neutrophils sticking to the endothelium. This may be an explanation, but we don't have that kind of access to see what's going on sort of in the choroid, unfortunately.
Beautiful talk, a perfect answer. Thank you.
Hi,
One more
Pat.
question.
Hi, Pat. It's Bela Anandapte from Kola Institute. So I was actually, as you were talking, thinking about the choriocapillaris and the glomerular network of vessels, both highly fenestrated, and both, you said, have fairly thick glycocalyx. And in my mind, I started to think of it as a protective layer. Why do these vessels that are highly fenestrated not leak? So is there any regulation? I mean, you answered the question about fenestrations, about does the glycocalyx regulate actually fenestrations, or when you lose the glycocalyx, can these vessels be more leaky? And the second part of the question is there a way to image and measure glycocalyx as a whole? I know it's a combination of lots of things, but if you had to say, is the glycocalyx changed in thickness in AMD, for instance, or in patients with, choroidal neovascularization, or was there a way to do that?
Yeah, so first part of the question, I would say the glycocalyx in our hands definitely regulates fenestrations, because in the knockouts that we've done, and in fact, even when we did the injections of the RNA, we could see reduced fenestrations. Let me think through this. Well, you need VEGF-R2 signaling to maintain fenestrations. That's well known. So people have even shown in primates that when you put aflibercept into a primate eye, the fenestrations go away temporarily, at least. So you do need I would say, an intact glycocalyx to signal, even just in the early studies of the knockouts, we measured just choroidal thickness. Very hard, as you know, to get at the choroid, but we measured choroid, and it was much thinned. And when you have choroidal thinning, it's usually because the choriocapillaris has dropped out, not because the larger vessels are gone. So lack of endomucin definitely leads to choroidal thinning. Again, back to measurements, I think I think we just have to get initially just really good at doing, producing a photo like I showed you in the first one, where somebody actually measured the height. And again, we're collaborating with Kevin Chasey at Duke right now to see if we can come up with a model to quantify it. So what we've done is taken human retinal capillary endothelium, isolated the membranes, and animals with just SI scrambled and SI endomucin, and he's going to compare those to see if we can even detect the difference between glycocalyx that has endomucin and what doesn't, and then also hoping that we might back into being able to figure out what percentage of the at least cultured retinal endothelium, which is probably not the same as in vivo, glycocalyx is composed of endomucin. So not an easy thing. I mean,
Thanks, Pat.
no direct way.
That was great. Thanks a million, Pat, for joining us. We're going to move on to our next speaker.